Hello, my lovely physiologist, Michele here. We're back with chapter 12, and now we're ready to pick up a few more details about that action potential. What we're picking up on here is the idea that the action potential is unidirectional. What we see when we draw out our neurons, so if we draw our multipolar motor neurons, so draw that cell body as like a circle with a nucleus, maybe draw those tree branch-like dendrites coming off of the cell body, and then that one super long axon with the axon terminal and the synaptic knob. What we see is that the action potential can maybe, come in from the dendrites to the cell body, and then we actually see the stimulus picking up at that location that we call the axon hillock. And then we see it traveling down the axon until it reaches the synaptic knob. Right? And so when we talk about the action potential as unidirectional, we're saying it's going to go one direction only from the axon hillock to the synaptic knob. And this is because of the refractory period. So it's allowing for, or creates, maybe we should say, the unidirectional movement of the action potential. Maybe creates is bad. Maybe we should say responsible for. Something along those lines. Now, I like to describe the refractory period as where it's either impossible to stimulate the cell again or super duper hard. So my personal definition for a refractory period is that it's either impossible to stimulate the cell again or is super duper hard. So when we talk about this, we'll say then that there's two types of refractory periods. We have what's called the absolute refractory period, and then we have the relative refractory period. And so the absolute, I would say, is, and I'm going to use quotations here, it's impossible to stimulate the cell again. Whereas in the case of the relative, I'll use quotes again. Here, I'll say super duper hard. That's my technical terminology, y'all. Now, why is it impossible to stimulate the cell again during the absolute period? Because what we'll see is that the voltage-gated sodium channel is going to be deactivated. You can't get an action potential if you can't get sodium into your cell. The only way to depolarize a cell is to flood it with sodium, or that's the most common way to get the cell to be stimulated, is to flood it with sodium. So if you're not able to flood the cell with sodium, you're not able to stimulate the cell again. In the case of the relative refractory period, you have voltage-gated potassium channels activated or open, and so you have to overcome all of the sodium that's leaving. So more sodium would be required to get to threshold. So relative is super duper hard because you have all this potassium that's in the process of leaving the cell, and you have to kind of fill in that gap plus also the sodium that's required just to take it to threshold anyway. So because of this refractory period, it just basically pushes the action potential in one direction. Let's take a look at the graph that we drew in our last video. So we're looking at graph, you should already have this in your notes. So we have the x-axis is time, we have the y-axis is the membrane potential, and then we graphed out our action potential starting at resting membrane potential at -70 millivolts, reaching threshold at -60 millivolts. That triggers that depolarization of the action potential. So we bring our line up to a peak value of +30 millivolts, and then we see it start to repolarize, meaning heading down towards -70, dropping below that to -90, and then being restored to resting from the activity of the sodium-potassium exchange pump. So what we're looking at here is essentially, I'm going to draw kind of a line down from my peak value heading down towards my threshold and my timeline. And what we're seeing is all of this time under this graph, under this line, is our refractory period, and everything that I have kind of on the, let me redraw this line. So we're going to actually come over a little bit, maybe about here. We have our absolute refractory, and then we have our relative refractory period. And so what we're seeing is, you know, while the cell's being stimulated and those voltage gated sodium channels are open, all the amount of sodium that can flood the cell is already flooding the cell. And then we'll see that those voltage gated sodium channels deactivate at plus 30, and they stay deactivated for a while. And all that period that they're deactivated, that's the absolute refractory period. And then we move into the relative refractory period when those voltage gated sodium channels can be stimulated, but it's going to be super duper hard to actually hit threshold when you actively have potassium leaving the cell. Now, this takes us directly into the conversation of propagation. And propagation, I like to sometimes use the description of travels. So this is how the action potential travels an excitable membrane. And so this mechanism is going to be true whether we're looking at the sarcolemma, whether we're looking at the T tubules. So remember, that's from chapter 10. Or whether we're looking at an axon, or if we're looking at a cardiac muscle, smooth muscle, and so forth. So if you have an excitable membrane, then your action potential can propagate along that membrane. And so this term propagation means steps are repeated. So once you have the pattern and the sequence of steps, then it happens again and 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 again until you run out of excitable membrane. So that's what we mean by propagation. Now there's two different types of propagation. We have what's called continuous, and then we have what's called saltatory, where saltatory literally translates to jumps, jumping. In the case of the continuous propagation, this is happening on unmyelinated axons. So we're going to see this primarily in the central nervous system, where those axons are, or those neurons, I should say, are clustered together with real close proximity in the brain. And so in the case of the continuous propagation, what we see is we get the action potential generated at the axon hillock. So that's everything that we already know. When we see the stimulus being received at the axon hillock, that's opening up those voltage chemically gated sodium channels. That gets us our sodium team A into the cell. That initial depolarization takes us to threshold, which opens up the voltage gated sodium channels. And the key thing that we see happening then is that the sodium is going to spread inside the axoplasm. Axoplasm, remember, is the cytoplasm of the axon. So the idea here is, if I'm drawing kind of like a tube to represent my axon, I have this sodium coming in. I have sodium coming in from those chemically gated sodium channels initially, or then those voltage gated sodium channels. And then it's just going to spread along the axon. So sodium doesn't like come in and stay in place. It comes in and it spreads. And that spread is what we call local current. So the spread of the sodium inside of the exoplasm is described as local current. And what that's doing then is from the inside, taking the membrane to threshold. So from the inside, takes the next section of membrane to threshold, meaning opens voltage gated sodium channels. Remember, the moment you have threshold, you have your action potential. So if you're taking that next section of membrane to either the negative 55 millivolts or the negative 60 millivolts, you're going to get your action potential, right? So this is that idea of when we started telling the story and we said you got to get sodium team A in, and once you get sodium team A in, that triggers sodium team B to come in. So think of that sodium team A as coming into the cell, opening up those voltage gated sodium channels, and then you get more sodium coming in. As more sodium floods into the axon, it moves just through the process of diffusion from a high to low concentration. Is taking that next section to threshold, boom, that means it's opening up those voltage gated sodium channels, and you're continuing all the way down until you run out of axon. Now, in the case of the saltatory propagation, this is happening with your myelinated axons. So everything in the peripheral nervous system is going to be myelinated. And then obviously you're going to have some myelinated axons that are in the spinal cord and the brain as well. So we'll see myelinated axons and saltatory propagation in both central nervous system and peripheral nervous system. Now the idea here is that we're going to be jumping. And so this is faster. Instead of having this process of sodium coming in, opening up voltage gated sodium channels, spreading, coming in, opening up voltage gated sodium channels and so forth, we see that we're only going to take nodes of Ranvier to threshold. So that means the sodium can come in, spread through those myelinated sections, and then reach an exposed piece of axon, which is the node of Ranvier, open up those voltage gated sodium channels. Sodium can come in, sodium spreads through the myelinated section, that's our local current, and then it's going to take that reach to that next node of Ranvier, and then it's going to just do the threshold there, opening up those voltage gated sodium channels and so forth. So it's a repeat. Okay. Stay tuned for our next video. We are nearing the end of our chapter 12. As always, take care of yourselves and each other.